Our macromolecule odyssey continues with part 3, the proteins. So, proteins, we are back to our monomer-polymer rules. So, we're free from the lipids and their evil refusal to obey monomer-polymer rules. What are the monomer-polymer rules for proteins? Well, that's pretty easy. The monomer of a protein is the amino acid. Right? The polymer of a protein is a bunch of amino acids chained together. So poly. And then before, in order to make this next part make sense, I'll tell you about the bonds between monomers. But first off, monomer is an amino acid, blah, blah, blah. I'll get to that in a second. The polymer is a polypeptide. Why is it a polypeptide? Like, why isn't it a polyamino acid or something like that? Well, the answer is pretty simple in that if you look at some amino acids, circle, triangle, I don't know, hexagon. When you covalently bond them together to get a chain, the special name for those bonds is peptide bonds. So the peptide bond is the covalent bond that links two amino acids together to form a chain. So a polypeptide is literally many peptide bond joined amino acids. So polypeptide. In the cellular world, especially in human or mammalian critters, there are more than 20 different kinds of amino acids that are generally considered biologically important for making the grand majority of your proteins. So, will you have to memorize them? No, obviously, right? When they come up much later in transcription translation, you'll actually be given a table to reference. So, but there are more than 20 different types of amino acids present that you can chain together into hundreds or thousands of amino acid chains in order to get something cool and functional. So, speaking of functional, what are the functional groups on an amino acid? Gee, I wonder if perhaps an amino group could be present. And then how about functional groups for the second one, acid? Well, so far, every time we've talked about acids, it's been carboxylic acid. So, carboxyl group or carboxylic acid. So there you go. That's pretty easy. If it's a carbon compound that has both an amino group on it and a carboxyl group on it, it is an amino acid. Whee. Now, you might notice the word polypeptide is present, not the word protein. And there's a reason for that. Because a protein, the term protein is given to something with a biological function, right? So protein is at the biologically active or functional level. Oops. I want active. Biologically. Active or functional. Right? In other words, the protein works. Is doing its thing. A polypeptide is not a functional protein. proteins have to undergo a very specific three-dimensional structuring in order to become biologically active and functional. And we call these the 4 levels of protein structure, of which 3 of them must be met in order to get something biologically active, and then some proteins get to the 4th. So, the first level of structure is pretty simple. Primary structure, also 1 degree, first degree structure, and it's literally the polypeptide chain. So, that's pretty simple. Ta-da! That's primary structure. That's the first level of protein structure, literally the polypeptide chain that is produced at the ribosome. So, not a big deal. I mentioned ribosome. I'm going to mention ribosomes in part 4, but ribosomes, since I'm saying it offhand, are the site of protein synthesis. Basically, if you need proteins, they're getting made at a ribosome. So, they're like the cellular machinery where proteins are made. So, polypeptide chain, just fresh off the ribosome. Secondary structure is hydrogen bonding. So we have hydrogen bonding betwixt hydrogen and, amazingly enough, electronegative atoms in the amino acids. Right? So you get hydrogen bonding, and interestingly enough, these hydrogen bondings produce 2 basic shapes. The first is a slinky. Right? And so you got your hydrogen bonds that hold the slinky shape together. So, the slinky, or, since we're very fond of it, the helix. And since this is one of 2 options, we'll be real science-y and call this option alpha. So this is the alpha helix. And then option 2 is a sheet of paper folded accordion style. Ta-da! See that? What does that look like? Well, you got your chain, and then you got your accordion folds. And you've got hydrogen bonds holding them together. To hold that accordion folded paper form together. Ta-da! Like this. So, you got the accordion, and your amino acid chain comes in here, and then comes around there, and comes out there. So, if you're a fancy 50s person, you might be pretty proud of the little folds in your pants that go all the way along, and those would be called a pleat. A fold is a pleat. So this is a sort of 2-dimensional pleated structure. So perhaps a pleated sheet. Option 2, or option beta, the beta pleated sheet. Or accordion style. The accordion fold. Not too bad. That's secondary. So, ta-da! We've got the alpha helix and the beta pleated sheet. Primary, very simple, polypeptide chain. If you know the name of the polymer, then congratulations, you know primary structure. Then slinkies and accordion folds, or alpha helices and beta pleated sheets. Hydrogen bonds, beta, blah, blah. And there's a polypeptide. And then here's, obviously, with much worse artistry, a beta pleated sheet and a alpha helix. So, not too bad. All right, we're not done. Third level structure is where a lot of proteins become functional, biologically active. Third level is kind of hard to describe, so we kind of just, so first off, did I put down 2 degree? Secondary, abbreviated with 2 degree often. So tertiary, amazingly enough, 3rd degree structure. We just call this the compact ribbon. And basically what happens is we get disulfide bridges between amino acids. These are not peptide bonds, right? Not peptide bonds. Basically, what's going on here is we're forming stronger covalent bonds in order to hold your your protein into a much more complicated, large, advanced, and very specifically shaped 3-dimensional structure. So, right, it's a complicated 3-dimensional structure, and the reason here, well, first off, disulfide bridges, disulfide, so basically you have a sulfur-sulfur covalent bond. So if you have amino acid square over here, and it happens to have a sulfur, and then amino acid circle over here, and it's one of the ones that has a sulfur, then they can form a disulfide bridge. And it will hold that little section together. So, ta-da! Okay, so what's going on with this compact ribbon? There's a few things going on with this compact ribbon. First off, there are hydrophobic and hydrophilic amino acids. So some amino acids are hydrophobic. A lot of amino acids are hydrophilic. And during tertiary structure, hydrophobic amino acids get shunted to the center. So they get sent to the core of the of the protein. Sorry. So hydrophobic amino acids get shunted to the core of our protein, where they are protected from the water. And amazingly enough, hydrophilic amino acids go to the outer layers, or make up the outer layers. So this is a big thing that happens here. And then there's a variety of important structural shapes these things take. 3-dimensional shape is extremely important. So first off, little vocab, 3-dimensional shape or structure. In other words, how it's put together in 3D space. This is called conformation. Right? In other words, tertiary structure is all about forming this large, complicated, compact ribbon in order to get the conformation for our functioning protein. And as in most aspects of biology, form equals function. The conformation of the protein will determine its functionality. If its conformation doesn't shake out correctly, the protein won't function correctly. So form equals function. And the compact ribbon, the tertiary structure, is where we're going to get most proteins becoming functional. So here you can see we've got some of our secondary structure things in here. And then we've got our disulfide bridges or bonds holding these things together. And it becomes this very complicated structure. That's tertiary. One more to go. Quaternary or fourth degree structure. This is not universal to all proteins. What makes quaternary different? Well, if things are mostly active at tertiary, quaternary isn't about further folding a protein or a polypeptide in order to make it functional, more functional. What it is covalently bonding multiple tertiary polypeptides together. I'll do two more because it's pretty. There's my other tertiary, and then here's my last tertiary, and I went for 4 because it's quaternary, and that makes my brain happy. So here we have multiple tertiary proteins covalently bonded together. Not too bad. Multiple tertiary structured proteins put together. Here you can see much worse artistry, but an example. So we have 1, 2, 3, 4 tertiary proteins put together. This is a great example of a quaternary protein. And this one happens to be loaded up with a bunch of heme, which is great at bonding to iron. So heme, which has iron. I wonder what protein this is. Give it a thought. And while you're thinking, I can give you a hint. Iron, this positively charged iron, does a pretty good job of attracting oxygen. So heme attracting oxygen. Ooh. It's a protein on a cell in your body. Ooh. What's that? Did I hear someone say hemoglobin? That is exactly what this is. Hemoglobin. Hemoglobin is a quaternary protein example, and it's for transporting oxygen. So it's the protein that's extremely dense on red blood cells, and it transports oxygen, or rather, the red blood cell transports oxygen. Heme groups on the hemoglobin bind to oxygen. So, the billions of hemoglobin proteins packed into a red blood cell are what allow a red blood cell to bind to oxygen and efficiently transport it through the body. Okay, so quick incomplete list of protein functions and examples. Enzymes, this is a big one. Enzymes is a big one. Enzymes are considered a catalyst. A catalyst is anything that increases a reaction rate. For instance, one of the big chemical reactions we'll talk about is this. 6O2 plus C6H12O6 yields 6CO2 plus 6H2O. This is a chemical reaction that we'll talk about a bunch. A catalyst is anything that increases the rate of this reaction of 6 oxygen and a glucose being broken down and reformed to get 6 carbon dioxide and 6 water. Catalyst increases the rate at which this reaction occurs. Enzymes are biological catalysts. We've got a whole thing on enzymes, so this is a very brief thing. And enzymes are proteins. All right? Fun, simple rule on enzymes. If it ends in ASE, it's an enzyme. All right? So, not too bad. Enzymes are a very big deal. We have a whole lecture on them. So, increased reaction frequency, ASE, and they're often named for what they do or what they act on. So, not too bad. There we go. Amylase in your saliva breaks down amylose. Starch is called amylose. So amylase, ASE means enzyme, breaks down amylose because OSE means carbohydrate and OSE is starch. So there you go. Reverse transcriptase does transcription in reverse. DNA helicase is involved with unwinding the DNA helix. So not too bad. And that's proteins. So hooray. Pick up on part 4, nucleic acids. Have a break, eat some candy, whatever.